So welcome to the third webinar of the VALITEST webinar series on the concept of test validation in plant health. So after the first webinar, which was on the importance of validation, and the second one on how to adopt a new test, today we will speak on the use and validation of on-site tests. So we have two speakers with us today. Denise Altenbach is our first speaker. She is a scientific director of BIOREBA and head of the testing service laboratory with more than 10 years of experience in developing, producing, and commercializing diagnostic kits. And she will present you the experience of a kit producer in the use and validation of on-site tests. And our second speaker is Jean-Philippe Renvoisé. Jean-Philippe works at the quarantine unit of the plant health laboratory of ANSES in France, and he will present you the experience of VALITEST diagnostic laboratories on the validation of on-site tests. So with this said, I will give the floor now to Denise Altenbach, and she will present you the introduction and also the point of view of kit manufacturer.
Thank you very much, Charlotte. So I will start with the introduction. And the introduction is, first of all, I would like to show you what are on-site tests. So an on-site test is the fast assay where you get results within a few minutes. It's optimal test for single testing. The handling is very easy and it's especially suitable for testing on-site, so in greenhouses or in the fields. There is no need for special laboratory equipment. You see here how a field inspector got organized, so he has everything ready to perform a test in the field. Please note that there are on-site tests based on molecular methods as well. They are based on isothermal amplification, but today's seminar is restricted to on-site tests based on serological methods. So what do you need to perform an on-site test? First of all, you need symptomatic tissue. Then you need a test, like this is shown here as a complete kit. And then what you need is a test user. This is optimally trained personnel, and they should be trained for sampling and performing the test itself. We will quickly show you how such an on-site test, the AgriStrip, is working in a video. Sorry, I was muted. So the sample is first in your extraction bag. Then you grind a little bit after having added the extraction buffer. You can take a few drops from the bag, transfer it to a cuvette. Then a test strip is entered. And then you start seeing how the liquid, so your sample, starts migrating through the strip. And after a while, you see lines appearing. This is usually after a few minutes the case. The upper line is the control line, the lower line is the test line, and this upper line shows you that the test worked. Only one line is the test is negative. If you have two lines, the test is positive. So when and how do we use on-site tests? You first have to know about your need and the context of use. So you should reflect what is the type of matrix you want to analyze. Is it, example, leaves from your plant? Are these leaves symptomatic or not? And are there restrictions like the testing season, that you have an optimal season to test? Then you have to check, do my needs fit with the intended use of an on-site test? So you should check performance characteristics and intended use of the test. This information can be found on the manufacturer's product information. You just have to identify potential modifications, and then you should do a small risk analysis and decide if you need additional validation or if verification of your needs is sufficient or your modifications. Then it's always important to ask the question is if the end user should be trained, and please always ask us for advice. We know our tests and we are happy to assist you in any case. This is to show you how a product information looks like. Here you find, as I told before, a lot of information already about the intended use, optimal sampling, etc. So we come to some first questions for you. You will have to answer them just after. The first question will be, have you already used an on-site test? You can answer just simply yes or no. And the second question will be, have you already tried to validate an on-site test?
So almost 90% of the people have voted, so I will close the poll. So Denise,
I don't see the answers, Charlotte.
Oh, really?
Yes. Now I see it. Thank you very much. So 43% of people in the webinar already used an on-site test, 75 didn't use one yet. Okay. The second.
So yeah, I will put the second one. So here again, we are close to 90% of the people that have voted, so I will close the poll.
So 19% of the attendees already have tried to validate an on-site test, but 89%, so the majority did, 81% did not. Thank you. So I will come to the main part of my presentation. It's about the use and validation of on-site tests, and this is in the view of us as a manufacturer.
Wait, Denise, you're not sharing your screen. Yeah, it's okay now.
Okay, so I will talk about the manufacturer's view on validation of tests. First of all, what do we do when we decide that we want to produce an on-site test? we have to select, first of all, antibodies that will be the basis of our tests. For the Plumppox agri-strip, we decided to take into account or to use antibodies that had been validated in extensive collections of virus isolates from all over the world with different partners. And these antibodies had proven to be very efficient in ELISA tests and very reliable as well. As a second test, then we were checking if these antibodies were functional in lateral flow device format. And this is not obligatory the case. Sometimes we have very nice antibodies that work nicely in ELISA, that are perfectly specific and have a high sensitivity, but then they don't work in lateral flow format. And to understand this a little bit, I would like to show you how a lateral flow test is working. So here you see a lateral flow test. In the middle, you have antibodies applied that are for the detection of the pathogen of interest. And also in the basis of the test here, you have colloidal gold particles that are also labeled with antibodies specific for the detection of your pathogen of interest. So these gold particles are sprayed to the bottom of the test, and now if your pathogen, shown here, is present in the extract, the liquid, you enter the strip into your extract and the liquid starts migrating through the test. So once your pathogen of interest comes into contact with these gold particles that are labeled with the antibodies, it will bind to the antibodies. And like a small package, so there's no social distancing there, like a package, this will flow through the strip. And once this comes to the test line, where there are the other antibodies that will recognize the pathogen, this all package will bind here. And since this colloidal gold is of reddish color, you will see color development at the test line. In the test line, in the control line, you have a species-species interaction of antibodies, and there's always an overload of antibodies or gold labeled with antibodies. So this line will always appear. So an antibody that is used for a lateral flow test needs to be membrane compatible, and also you need to couple gold particles, and still the accessible sites for the pathogen have to be still there. So how is production done? To show you, here we produce in card format. Here you see a membrane attached to the backing card, and we apply the antibodies. Here you see the glass fiber where the colloidal gold was sprayed to. And finally, we assemble all components of the card. And last but not least, they are cut into strips. Now for validation of such a lateral flow test. For us as manufacturer, validation and protocol optimization is a process that goes hand in hand. The main criteria we take into account are analytic sensitivity and analytic specificity. And once this is satisfactory, we decide to continue. If it's not satisfactory, we stop at that point. But if it's satisfactory, of course, we take other criteria into account, which are repeatability, reproducibility, stability, and of course, also lot-to-lot consistency in the end when it goes to manufacturing. So about the main validation criteria, analytic specificity. For the example of Plumppox virus, we wanted to have detection of all Plumppox isolates known to date and to have no cross-reactivity with other viruses occurring on the same host. Also very important for us is selectivity. Selectivity means that there is low or no background with healthy host tissues. So we wish to have low or no background with healthy leaves or petals of prunus species in that case. Then analytic sensitivity, we also look at that, but not at the cost of selectivity. So for lateral flow test, it's always, we have to find the balance if we have high analytic sensitivity and low background. There's also always a balance to take here. And I would like to explain you why. So for lateral flow this test, there is a slight difference to other diagnostic tests. In a PCR, you always have in advance, you have a purification of DNA or RNA, and in a laser test, you have a wash step. But the lateral flow has to deal directly with the sample matrix. So this sample matrix is directly flowing through the test, and this is quite a challenge in development. What is the playground for optimization? So it's actually the factors that will also impact on sensitivity and specificity. Our playground has two aspects. The first one is the formulation actually of a lateral flow test strip, where we can decide or play with the antibody mix. So which antibodies do we couple to gold, which ones do we couple to bind to the membrane, and so on. And then also the concentration of and ratios of these antibodies is our part of our playground. This is BIOREBA's job. And the other important part that has an influence on test performance is sample preparation. Here we as manufacturer do a lot of work on sample extraction buffers. So we work and optimize buffers. The sample dilution is very crucial for lateral flow tests. Then the extraction procedure itself. And then, of course, the use of the recommended sample tissue, so the matrix. This is a joint job we are doing with the end user of the strip, but we also give support with the buffer and the dilution that we recommend. Now comes another movie where we show what is the optimal sample extraction.
Denise, you have to unmute yourself.
Yes, sorry, I didn't see it. So the recommended sample size for Plumpbox virus testing is about the, sorry, I was again muted. It's a €2 size is recommended piece. It's about 8 square centimeters, and this is what you see, what I was cutting here. So we take the phloem tissue basically of the basis of the leaf, and we add this piece of leaf into an extraction bag. This extraction bag has an intermediate layer, which is semi-permeable. So if you place it in the bag, afterwards, you will be able to pipette from the front without the pipette getting blocked. So we add now extraction buffer B, 3 milliliter, and this results in then a dilution of 1:20 to start with. We briefly mix and homogenize here with a handheld homogenizer. So this has not to be 100%, but you have to crush or squeeze most of the sample. You can also use the back of a pen or something, and this is how then the 1:20 dilution looks like. The final dilution, as we recommend, it needs to be 1:40 for Plumpbox virus. Here we use again the extraction buffer B and do now a dilution directly by adding 2 drops of buffer first into a cuvette. And then we take 2 drops from our 1:20 diluted sample to have the optimal dilution of 1:40. We put the liquid to the bottom and are ready to insert the strip. So thank you. Do you see my screen again, the presentation?
Yes, Denise.
Very good. So to summarize, we have an optimal sample dilution of 1 to 40, which you see with the yellow flash. It's plumpox, and also on the right side for healthy plant sample, 1 to 40. We would like to show you here what happens if you didn't dilute sufficiently. So what is happening with a 1 to 10 dilution, or how it is if you dilute too much, 1 to 120. So we inserted strips in these cuvettes, and after 7 minutes, we see the result already appearing here. You have the optimum 1 to 40, where you see a very nice test line and the control line, only the control line for the healthy sample. Now you see the test line in the too diluted sample is much less pronounced, and also the line is less pronounced for the too dense sample. And here especially you can encounter a problem because the strip can get blocked. You see this very greenish plant sample remainings at the bottom, and this can completely block your test. So the results from our validation and optimization for plumpox agristrip were that we detect all plumpox isolates of this collection we had, and then there was no cross-reactivity with other viruses. We also checked that, like for example PDV or PNRSV, so viruses that occur on the same host plants. And then we had optimized also to have low and no background with healthy host tissue, and this was achieved by use of the optimal extraction buffer and by the use of the recommended sample dilution and sample size, which influences then, of course, the dilution. We as manufacturer always try to have an extended validation, and this includes, if possible, a verification with external partners. We try to work together with reference laboratories or with laboratories having access to a nice virus collection. And in the case of the plumpox virus agristrip, we used as verification with external partners. We collaborated with the Agricultural Institute of Slovenia, so with Mojca Mann and Irena Mavric, and they did a nice study and compared sensitivity of field tests, and they compared serological and molecular techniques. And the main results were that they could detect all the isolates, also with the agristrip, that are present in Slovenia, and they showed nicely that flowers of apricot and plum in full bloom were a very nice source for good detection of plumpox virus. They also nicely show in this publication that the use of symptomatic plant samples for the agristrip is essential. And then if whenever possible, we are happy to participate in proficiency tests. And Jean-Philippe will, Renvoisé, he will just talk afterwards about his experience in the frame of the VALITEST project. Once we consider development and validation from the manufacturer view finished, we define the scope or intended use of the test. And so this is based on the results we obtained, development, validation, and optimization. And the main results for the plumpox agristrip are that fresh tissue is the optimal tissue in the recommended test season. Then we recommend to use symptomatic tissue. And in order to show you this a bit better, you see here results of one leaf. On the left side, we were analyzing left part of the leaf. On the right side, where you have much more symptoms, we have the test here. And you see that the difference in signal intensity is quite enormous. So with the same leaf, you can get quite different results. So symptoms are quite essential. Then we recommend the use of extraction buffer B and a sample dilution of 1:40. Now we want to have a small insight into production and standardization, and also in customer training. So since this Plumpox agri-strip is part of an existing product line at BIOREBA, we have this product line since 10 years, or more than 10 years. We can use for production and quality control standard operation procedures. So we have all our quality control protocols available. The criteria that we are taking into account are stability tests, shelf life, storage conditions, and also lot-to-lot consistency. And then also we are very happy to assist training of users, but please remember that the protocol and the test, the lateral flow test itself, it's half of the test. The other half is sampling and sample extraction, and the user should also be trained for this. So I would like to conclude. We have 3 parameters for a successful test. This is a validated test, appropriate sampling, and trained test performer. And now I would like to give the floor to Jean-Philippe Renvoisé, and he will talk about what are the challenges for a successful evaluation by the user. Thank you very much.
So Jean-Philippe, you should be able to share your screen now. Yes, it's working. Jean-Philippe, you're muted. We cannot hear you. Yeah, you can speak now.
Can you hear me? So this part of the webinar will present you the user view regarding the validation of the on-site test. By the user, you should understand an organization or a person who has an on-site validation project. The objective of this presentation is to identify the specificity of on-site test validation, but not to describe the whole process of validation. Here are the different items I will address in this webinar. These items are presented as advices for the user. First, you should identify the performance criteria to be evaluated, then to choose the appropriate evaluation, and identify parameters that can interfere with the evaluation and validate the modification made. And finally, to conclude on performance characteristics. To make the presentation more clear, I will use examples taken from the test performance studies, which included on-site tests and different opinions from TPS organizers. Of course, that been organized in the framework of the VALITEST project. So first, we begin by identification of the performance criteria to be evaluated. This should be based on the intended use of on-site tests. And the intended use has already been presented by Denise Altenbach. Here you can see example of expected performances for on-site tests that you could describe as, for example, detection of high and medium target loads, positive results for isolescent strains, negative results for non-target pests, and negative results for healthy plants. The expected performances for on-site tests are linked to performance criteria. For example, analytical sensitivity, inclusivity, exclusivity, which combines with inclusivity to be calculated as analytical specificity, and finally selectivity. Some are considered as most important for on-site tests. Here you can see that analytical sensitivity is not recognized as most important on-site tests. And you can notice that both users and manufacturers have almost the same point of view regarding this criteria. Here are some other performances that you could expect for on-site tests. So positive results with positive samples, negative results with negative samples. You can also expect confidence in one result per sample, confidence in results given different conditions, confidence in positive test results, and confidence in a negative test results. The corresponding performance criteria are diagnostic sensitivity, diagnostic specificity, repeatability, reproducibility, and the two likelihood ratios that can be calculated with the previous performance criteria. Here you can also see, as a common view with the manufacturer, that repeatability and reproducibility are not considered as most important. Then you can choose the appropriate evaluation. And you have the choice between two main types of evaluation. First, you have the in-house evaluation, which is an internal validation project. For example, it can be the preliminary study of a TPS, but it can be totally independent. And second, you have the test performance study, which is an interlaboratory evaluation, which involves a TPS organizer and his team, and also participants of the TPS. You have some different things regarding in-house evaluation and TPS. In in-house evaluation case, the on-site conditions can be respected, the complete protocol can be easily evaluated, and you can also have the benefit of a high number of different samples. For the test performance study, you will produce a high number of results, so it will give you a high confidence in performance characteristics, and it's of course suitable for reproducibility. As main weaknesses, in-house evaluation has a limited number of analytical environments and of course a limited number of operators, whereas test performance study has a limited number of different samples and it can be difficult to reproduce on-site condition. If we look at the specificity of the on-site test, of course, we can see that in-house evaluation is a little bit more appropriate because on-site condition can be respected, whereas it's more difficult to reproduce on-site condition in the case of the test performance study. Of course, both can supplement each other. And in the case of the VALITEST on-site evaluation, all TPS organizers systematically organize an in-house evaluation, so-called preliminary study, and the TPS itself. Then you should identify the different parameters if the conditions are too different from on-site conditions. And you have to consider protocols, samples, and operators as a possible source of deviation. In some cases, you may need to modify something, for example, modify the protocol, and so you will have to check that the modifications made have no influence and or validate the modification with the manufacturer. I will begin by the protocol point of view. I took the example of course, the same example as Denise. It's a lateral flow device to be used on a fresh symptomatic matrix of a host plant. So you can see on the left the different steps of the protocol, where you will have the sampling, the grinding, the dilution. Well, I took the case of the agrostrip, who needs a dilution, but some other LFD protocols don't need this step. Then the migration, and finally the interpretation of results. The sampling should begin by the recommended matrix. In that case, you can decide to evaluate the complete protocol. Of course, it's more restricted to, let's say, to in-house, because it's much more difficult for a TPS to organize a complete protocol evaluation. Nevertheless, you have possible issues for plant material availability, storage, and homogeneity of lots of samples. You may also decide to evaluate partial protocol. For example, that's been done in most TPSs organized in the framework of the VALITEST project. It can also be suitable for the in-house evaluation. In that case, you have a homogeneity and matrix availability can be easier to manage. And as you can see on the different steps, you have plant pieces, matrix exact, and different matrix exact can be already diluted in the last case. And you can see that the sample preparation level is linked to the first step that you want to begin the evaluation. Of course, you have to use the recommended buffers and bags by the manufacturer. The advice that could be given is to be as close as possible to the complete original protocol, but you may have difficulties with the plant material availability. So the different TPS organizer in the VALITEST project choose different options depending on the metrics available. And the modification were validated either by TPS organizers themselves or discussed with the manufacturer. Regarding the operators, they can have an influence on the test results. I will begin by the in-house case. The selection of operators should give priority to the trained operators, of course, if you have in your staff. Otherwise, you should think to train your operators, which is highly recommended. In that case, you should contact the manufacturer. In the case of the TPS, the equivalent of the selection of operators is the selection of participants, where the main criteria considered should be the possibility to participate according to the time schedule of the TPS, and a quality assurance in place or a traceability system in place. And because the on-site test can be used by the users who are not experts with the test or the method, the proficiency of the users was not a selection criteria for on-site test. Of course, you can also imagine to train the participants, but it's much more difficult to organize. You can replace it by giving more precise instructions to the participants. And now regarding the sample, the first important constraint is to manage the fresh material availability. First, you have to collect and test in the right season or deseasonalize in dedicated facilities if you have. It's of course easy for an in-house evaluation, but much more difficult to test. Nevertheless, it's the best option for on-site test. Otherwise, you need to organize alternative solutions. So I mean to collect and store and check for potential matrix effect. You so have different storage solutions. You can freeze dry, or extract nucleic acids. On the case of the TPS organized in the VALITEST project, it was organized in winter. So most of the TPS organizers choose different options for storage. Here is an example of an evidence of a matrix effect. On the TPS, so freeze-dried plant material ground into fine powder was used. The preliminary study results were impacted by the matrix preparation. So the protocol adjustment could not be validated due to the unavailability of fresh material. The manufacturer was contacted and gave his agreement on the protocol adjustments made. And finally, an in-house evaluation using fresh material has been organized to confirm the TPS results. Another part, an important part, you should collect the recommended matrix to avoid potential matrix effect. You should also collect with symptoms. It can be, of course, for the target you want to study, but also for the non-target pest if you want to assess exclusivity. Of course, you should collect in the best period, but something which is very important is that the best period for symptoms may not be the same for all targets or non-target pests. Here is another example of a case of unavailability of symptomatic plants. It was the case was the asymptomatic or polysymptomatic plants were only available for non-target pests. They were finally used for the preliminary study, only to check for potential cross-reaction, but not used in the TPS. And so the results had to be confirmed with fresh plant material. Also, you can use asymptomatic plant material. Of course, if you want to assess selectivity assessment, you have to use healthy plant material. You can also use dilution if you want to assess analytical sensitivity. What can be specific for on-site tests is that if symptomatic plant material is required, such as for agrostrip, for example, you should prepare more moderate dilutions and prepare a specific panel for on-site tests. Another case should be if symptomatic plant material is not required for your on-site test, possibly higher dilution could be prepared. Now regarding the homogeneity and assigned value for the lot of samples. For most on-site tests, have high limit of detection, and some cases you can have possible uneven distribution of the pest in the matrix. So the homogeneity of the lot sample is a crucial point to be met for all on-site tests. Here is a new example coming from a TPS organizer. The question was how to provide enough homogeneous plant material for all participants. The case was the use of plant extracts spiked with different isolates instead of fresh material. Then the confirmation in the preliminary study that the suggested modification didn't affect the results and that extracts were homogeneous and stable. The manufacturer was contacted and gave his agreement on the modification made. So the idea is not to detail all the whole process for homogeneity, assigned value, and stability. You will have more information on the specific webinar, which is called Preparation and Dispatch of Samples. Nothing is really specific for on-site tests for the next steps, but I will just cite this. You have to test sample loss for homogeneity, to determine the assigned value for samples, to choose homogeneous sample loss for panel constitution. When you want to constitute your panel of samples, you have to do this according to the performance criteria that you have chosen before, and according to the results of the homogeneity study. And finally, for the stability, you should store dedicated samples in the same condition as for participants, set a short-term deadline for analyzing samples, test with the same method as for homogeneity, and choose stable lots for statistical analysis. Finally, you will have to conclude on the performance characteristics. First is the intended use. While the intended use, I choose always the same kind of example of a test which has to be tested on fresh plant material, using symptomatic or not for plant material, and of course to be tested with a recommended host plant. Then you produce the performance characteristics, which is equivalent to the production of validation data, and you will have to answer the question, does it fit for the purpose? And in this case, the context of the disease has to be taken into account. The final decision should consider new criteria to be evaluated in the field condition, and this is very specific for on-site tests. For example, the speed of execution, the ease of implementation, the traceability of the test results, and some other criteria such as cost and level of training for operators, which should be taken into account for this decision. As the last speaker, I will present the take-home messages from Denise Altenbach and me. So our first advice is to collaborate with manufacturer for many reasons. First is to fully understand the protocol regarding the samples, the implementation of the test, interpretation of results, and troubleshooting. You may have asked for more information, ask for the same lot number of reagents for the validation. You may also discuss about abnormal results if you have, and ask for advices in case of modification of the protocol. And of course, the main advice is to be as close as possible to the on-site conditions regarding the matrix, the season, symptoms, and protocols. And this is the end of my presentation.
So thank you, Jean-Philippe, and thank you, Denise, for the nice presentations. So now we have 15 minutes for questions. So if you have any question, you can ask the question in the chat, or you can also raise your hand and I will give you the floor. So there is one first question. Are you considering antiserum production for TOB or FV? So I think it's more for Denise.
Yes, yes. Actually, we are working on that. We are in the process of validation actually of a serological test. This is first an ELISA test we take into account, just to let you know that most probably most of the, at least for us it's the case, it will not be a specific test for the ELISA for TOBRV. It will also detect other tobamoviruses like tomato mosaic or tobacco mosaic virus, but we think it's still a very valuable test if you test during production in the greenhouses, etc., to monitor for the virus group in general, and then you can still add an additional test if you need to know exactly what it is.
So Françoise Peter is asking on which matrix will it work?
So most probably, this is still part of our testing, most probably we will recommend it for leaf material and the PCR for testing on seeds.
So is there any question? Yes. So there is a question, can only on-site tests be enough for disease identification, or it will need also a further molecular test for identification?
Should I try to answer? Or
Yes,
Jean-Philippe?
Denise or
So
Jean-Philippe.
since on-site tests, if it doesn't show cross-reactivity with other viruses, let's say, then a positive result is a very good indication, because the sensitivity of lateral flow tests is much lower than sensitivity of a PCR test. So a positive result is in general quite reliable. So I think it's sufficient, but if it's a quarantine pathogen, it's always recommended to confirm with another test.
Jean-Philippe, you want to add something?
Well, maybe it would depend on the case. For example, if you have a field totally contaminated with a pest and you already know it's you already confirmed in the identification of this pest, you may not use PCRs or other tests if you have a positive test result for on-site tests.
So there is Peter Bonan that asked the floor, so I will give him the floor. I saw he put the question in the chat, but it will be easier. So Peter, if you want to take the floor.
Okay, thank you. Can you hear me?
Yes.
Yes. Yeah.
Okay. Nice presentations. I was also expecting molecular on-site tests, but anyway, now focusing on the ELISA, I have a question. If you have on-site tests for virus A and it has a very broad host range, then you have to test, let's say, maybe many viruses for specificity. So how do you handle with that?
Is the question for somebody in particular?
I will put the question also in the chat. Maybe more for Denise.
Yes, so this is a very challenging job. If we have a pest that affects many different host species, their validation usually is very time consuming. You need to collect viruses of many species. And what we generally do is we try to validate as much as possible, but we give the information more or less what was tested on our product information. So you could check, for example, if we indicate it works for testing in lily or potato or whatever. And if you have a question, you can contact us. If you test a different host, sometimes we have additional validation data and do not present all of it. And yeah, so that's how we approach it in general. And then also to have access to other viruses and maybe also virus isolates. It's very challenging, yeah. It sometimes takes years to validate the test.
So yeah, and maybe I can just add that more information can be found on how to analyze specificity for different disciplines, including virus, and also on how to perform your risk analysis to define the criteria that you need to analyze during validation. So all this information can be found in the EPO standard PM798. So we have one question. Do you know on-site tests for insects? I don't know, Denise, if you are developing at BIOREBA on-site test for insects.
No, we have, so far we had only plant matrix. So, but maybe Jean-Philippe, you can comment on,
No.
Also not.
Never heard about them. Maybe it exists, but I don't know.
Yeah, I know about LAMP-based assays that exist to check at airports, etc. But I'm not an expert in that field.
So I can see that Françoise wants to take the floor, so I will give her the floor. Françoise Peter, you can unmute yourself if you want to speak.
Yes, indeed, Denise, you're fully right. There are some LAMP assay that can be done at the airport with portable equipment, and that's for fruit flies. That's for or mainly Liriomyza trips, as far as I can remember. So they are on-site tests available. I know that I don't want to give a precedence to one trademark to another, but for sure I know that Optigen has some tests available for insects. And they are also on-site tests also for nematode, if I'm not mistaken. So.
Yeah, we have also a question regarding whether there are LAMP on-site tests for viruses. I guess they are, but
yes, again, yes, and yes. Bacteria as well. So, for example, for potato, for sure they are on-site tests for viruses and bacteria.
There is another question, which is
Charlotte, if I can just add something. So,
yeah.
To answer Peter's question, so you cannot validate all the matrices. And Denise is perfectly right. When you have a pest which has a number of matrices, you will have to make priorities. And usually you start with the highest priority host. So you will start, for example, with tomato or potato, etc. And you rank your host list of the pest, and then you do the validation for these ones. Nobody is in capacity to do everything for everything. So you will have to make priorities. And then, as Denise was saying, it's going to be clearly, explicitly mentioned on the product sheet. So that's how it works. And yeah, you've raised an issue, Peter, but I'm afraid there is a limitation in capacity to do everything all the time.
So yeah, so there was a question from Simona, how to choose the real LC sample? Could the material, is it sufficient? She is asking if it's sufficient if the material is just asymptomatic. Maybe Jean-Philippe, you can answer this question.
Well, if the on-site test you try to evaluate says clearly that the plant material should be symptomatic, well, you shouldn't do that. Otherwise, the manufacturer said, okay, would say to you, you have validated, you have bad results, but maybe it's due to the fact that the plants are not symptomatics. So it's a very big deviation. That would be very difficult to manage. I don't know what Denise, if Denise you have something to add to this.
Yeah, if you need to do validation, I think you should have a positive sample and one negative sample. And if usually you have the host plants, you can do some collections. And if you get clear negative results on leaves without symptoms, I think you should rely on, because you don't have to do a full validation if you rely on what work was done by the manufacturer or if it was published somewhere or whatever. But a negative control and a positive control, I would recommend always.
And then we have one last question from Tanya. With a possible LFD test for bacteria, is it possible to use the test line for extraction of bacteria? So by this, she means isolation of the bacteria on media to have a kind of enrichment.
I have no experience on that, so I can't answer.
I don't have much experience on that as well, but we have some preservatives in the extraction buffer that will inhibit bacterial growth. But if you don't, let's say, if you don't wait too long, you should be able to get like a to get a more dense sample, let's say, and do a small extraction. But I have no idea or guarantee if they would still start growing. Probably they will. They are usually quite robust, more robust than we wish to. Yeah.
Okay, so we are almost done with the webinar. It's almost 3. So Jean-Philippe, if you can switch to the last slide, I would so first thank all the participants for attending the webinar. There will be next week a practical training session on the analysis of performance characteristics, but the registration is already closed. The next webinar will be on Monday, 1st of February, and we will discuss on how do companies handle quality control and validation of products, and how the European Plant Diagnostic Industry Association Charter will help in improving this task. So I thank you for attending this webinar. There will be a survey at the end of the webinar when you close it. If you can answer this survey, thank you very much. It's really short. It will help us to see how you like the webinar and whether we can make some improvement. So with this, I would like to close the webinar. So thank you very much again, and see you at the next webinar of VALITEST. Bye-bye.
Bye-bye.